Hi, I'm Dr. David Ashbrook. I'm an assistant professor here at the University of Tennessee Health Science Centre. I'm going to talk to you today about gene network, but also about including genetics in your studies, how to do that, where you can store and get that data, and how you can use that data to do analyses, whoever you are, if you have no coding experience at all, or if you're a serious data scientist. And I'm going to take a why, how, where, and what approach to that. So asking, why do you need to include genetics in your studies? How can you do that? Where can you get genetics data from that's already been produced? And what can you do with that data without ever having to leave your keyboard? So the vast majority of biomedical studies in rodents are done in a single genome. In a single genome type, usually some form of C57 black 6, in mice, usually in Sprague-Dowley, in rats, but that's ultimately an N of one study. You're looking at a single genome. How do we expect a single genome to generalize across all mice? And then we expect that to generalize across all mammals, including all 8 billion humans that are walking around the planet. We know that there's big differences even between B6J and B6N, and they are strange. They're a single genome, they're inbred. So how do we assume that it's relevant to all humanity? And we have good evidence of why we need to include genetic diversity. So even with things like gene knockouts, transgenics, genetic background can have a huge effect on the phenotype. So this is a study I really like from one of our collaborators, Zane Palmer. They did a couple of different gene knockouts on different genetic backgrounds and carried out sort of different phenotypic behavioral experiments. And this is just total activity in the open field, a really easy phenotype to measure. And if they'd knocked out this gene on the B6 background, they'd have said, oh, there's no effect of this gene on activity. It's nothing to do with the activity system. If they'd knocked it out on the DBA2J background, they said, oh, wow, knocking it out doubles activity. So this gene has a huge impact on activity. And that should terrify you. How many times have you had a phenotype and looked for if a specific gene was linked to it and had gene knockout and gone, oh, there's nothing to do with it. This gene isn't relevant. And that just depended on what genetic background they did the experiment on. Not only that, but adding genetic variation allows you to get at more interesting information, more interesting data. So if we look here, this is a series of paper by Catherine Krasrowski and her lab in the Alzheimer's disease BXD model. And that's a cross between the 5X-FAD Alzheimer's model, familial Alzheimer's disease, crossed to BXD strains. So I'll talk more about the BXD, but effectively they are all descended from crosses between the B6J and DBA2J parents. And when you crossed 5X-FAD to the B6 or D2, you got about the same amount of amyloid beta being produced. But when you crossed it to different members of the family, some produced almost no amyloid beta and some produced a large amount. So you get this genetic variation, which tells you more. They saw differences in things like age of onset, in specific types of cognition, in the physiological decline, before cognitive decline. And this sort of really maps what you see in human families. You'll have even people with the same mutation have different ages of onsets. They'll have different specific symptoms. And we want to really model that. So it's no wonder that translation fails when you're trying to use a single genome to model the 8 billion human genomes. We need to include genetic variation. So how can we do that? At bare minimum, include more than one strain. We can use outbred populations. So for example, the diversity outbred panel in mice. And this gives you a large amount of genetic variation, a large amount of phenotypic variation. The downside of that is you don't have replicable genomes. If you have a single individual that is a large outlier, it lives 2-3 years with really good memory, you can't go back and ask what was happening to that at 6 months? What would happen if I gave it a high-fat diet? Whereas using genetic reference populations, you have replicable genomes, you can go back to the same genome repeatedly. And I'm going to talk to you about recombinant inbred strains, such as the BXD mice. So these are just one panel in mice. There's many others, for example, the HXB, BXH in rats, and there's 140 of the BXD strains. What these are crosses between C57 black 6J to DBA2J. They're crossed for 2 generations due to recombination, and then each of these is inbred for at least 20 generations, and some of these are up to over 180 generations now, and each line is fully inbred. So every individual in that line is genetically identical. So you can have an arbitrarily large number of replicate genomes, but each line, each strain is a cousin to the other. Each is a unique mosaic of B6 and D2 alleles. And what this allows you to do is include genetic variation, because you can include all these different strains, but control it because you have replicates. And this means you can produce identical genomes in any environment, any age, anywhere you are. I can go back to data that was collected in 1973 and remap it using sequencing data that we collected for all of these strains a couple of years ago. I can go to behavioral data collected in the 80s and correlate it with new hippocampal proteomics data that was gathered this year and find new significant biology. It allows us to do quantitative trait loci mapping, so associating regions of the genome with variation in a particular trait. And it means that we have fully coherent and reusable data. So again, having this data that we can map together and reuse, and I'll talk to you more about that later. But what it ultimately means is we can look for gene by environment interactions, all these complicated things that we really need to understand the whole biological system. And this allows us to do systems genetics and what I call experimental precision medicine. We don't want to just say we have a DNA variant, it does something to behavior. We want to know exactly what's happening at every stage. We want to say this variant is affecting expression of this gene in this cell type, that's having an effect on this tissue or this connections of this system, this having an outcome of behavior, and that behavior interacts with environment and disease risk and sex and aging. And we want to know all these connections, all these interactions. Because ultimately what we want with precision medicine is I give you a genome, I give you all the environmental information about that individual and predict what's going to happen to it. What age is it going to develop Alzheimer's disease? What age do you want to start an intervention? If I put that person on a low-calorie diet, will they develop Alzheimer's disease later or earlier? And if we can't do this in a rodent model, how do we expect to be able to do this in humans? So how do we make this data usable? Well, a lot of data is evaporative. It's used for one study, a really specific phenotype is measured, the results are published, there's maybe a table and a figure, and then the data kind of fade away. At best, it's locked on a hard drive in someone's drawer. At worst, it's disappeared completely. So it's not just about gathering more and more bigger and bigger data, but making sure that data is useful and reusable. So we have this idea of fair data, findable, accessible, interoperable, and reusable, but we say fair plus because also we want it to be reanalyzable on the fly. We want anyone to be able to use that data at any time. And all of this depends on gathering high quality metadata. I want to be able to go back to your data in 50 years time and know exactly what you did, what you measured. And one way you can do this and where we can get this data is using gene network. So there are many other sources of genetic data in model systems, but I'm going to talk to you about gene network. You've seen it up here in all of my slides. And this allows what I call smart, coherent data, data that can work together over time and is reanalyzable and reusable on the fly. So anyone, even if you have no coding skills, you can go on here, use the user interface and get new information out about a phenotype of interest. There's a lot of mouse data on here, also human, rats, and many plant data, and basically any genetic data can go on here, and all the data is contributed to by investigators. So we're more than welcome for your data to be archived here and accessible by other people. But I'm going to talk to you about that BXD family a bit more. It's the largest data set we have, and over 10,000 phenotypes have been measured for this family. When I say phenotypes, it's anything from coat color to blood glucose to hippocampus weight to what year the breeding of that strain was started. And this is sort of a breakdown of those 10,000 phenotypes. You'll see that about half are CNS related. And that, again, that represents the interests of the investigators who are using the BXD for a long time. A lot of them were neuroscientists. So were able to give us that neuroscience data, but data on other systems and of other types are also building up. We have over 100 omics data sets. A lot of that is old array-based transcriptomics, but there is newer RNA-seq transcriptomics. There is proteomics, the metabolomics, methylation data, and that data is for omics data is for over 30 tissues. Again, there's a bias towards different regions of the brain, and that's CNS bias. We have many other tissues from liver, spleen, lung, etc. And then this allows us to get coherent data. And that means that any new data set or any data set in those tens of thousands of omics can be integrated with any other data. So let's use this brain weight data as an example. So you can just go on that first page of Gene Network, search for brain weight, and you'll get 119 results. And I've just picked this one, which is whole brain weight, adjusted for age, body weight, and dissection error. And we can then immediately pull up basic statistics and a histogram. And what you'll see is the DVA2J parent is towards the bottom of that normal curve, isn't the lowest strain. And the C57 Black 6J mother is on the high side of that distribution, but again, isn't the highest. And what we can do is then correlate that phenotype against any of the 10,000 phenotypes that are on Gene Network, all 10,000, or any of the gene expression data sets. So we could say, let's correlate brain weight against hippocampus exon data and see if there's any exons that correlate with brain weight in the BXD. And it'll load all those correlations, and you can have millions of R values. So, and sort of almost infinitely big correlation matrix. And we can do that for, as I said, 10,000 phenotypes by 20,000 genes and even more for exons. We can also do QTL mapping. So what regions of the genomes are linked to our trait. So again, this is the same brain weight. You'll see there is a large QTL on chromosome 5 with a minus log P of about 5, a small QTL on chromosome 12, and another one on chromosome 15. So we know there's at least 3 different regions of the genome in the BXD family influencing brain weight. So that was very quick. I could give you an hour on any of these, but in conclusion, why should you include genetics? Because they have a huge effect on all phenotypes. If you're not including genetic variation, you risk both false positives and false negatives. How? Use more than one strain. Use a recombinant inbred strains, use a panel of inbred strains, use outbred populations, use diallel crosses. Again, I could give you an hour's talk about this. It's really important to have good quality data and good quality metadata, somewhere that's stored and accessible and that anyone can use. And use the databases that are out there. For example, Gene Network. Again, we're more than happy to include your data on there. Where can you get access to this? As I said, there's several data sets: Mouse Genome Informatics, Mouse Phenome Project, Rat Phenome Project, and Gene Network. And Gene Network, it allows archiving your data. It allows anyone to download that data. And if you are good with coding, you can download the data and use whatever language, whatever analysis you want to do. It also allows reanalysis on the fly, just using that simple graphical user interface. So anyone, wet lab scientist who just wants to know what genes correlate with my gene of interest in which regions of the brain, or is my gene even expressed in that region of the brain? They can ask those questions really quickly and really easily. And what this allows is genetic linkage. So there's correlations to find gene-gene interactions and gene-by-variant interactions. And there's a huge amount of acknowledgements for this. As I said, all of those phenotypes, all of that omics data is contributed by researchers who use the BXD, and just a small proportion of the ones that I've worked with are shown here. But particular thank yous to Rob Williams, who sort of Gene Network is his baby, to Casey, Melinda, and Jesse, who maintain and our BXD colony here at UTHSC, to Piotr, Arthur, and Zach and their team, who maintain the Gene Network website and keep improving it. And a thank you to all of you for listening. And I look forward to getting your questions at the mini symposium.